So, hello, this is Lars Kästner speaking, and I also welcome you to today's webinar for the relevance of ion channel interplay. And I want to start to introduce a little bit this mysterious relevance that's in capitals in the title. The relevance indeed is the reason how Justina and myself are connected, because it's a EU-funded PhD training program, and it's entitled Regulation of Red Cell Lifespan, Erythropoiesis, Survival, Senescence, and Clearance. And the acronym of this also complicated title is relevance. However, relevance is far more than a simple training program. It's a very special training program. It has numerous collaborators all over Europe, academia, and companies. And as you see, you have also NANION as a partner in the Relevance Consortium. So what makes this Relevance so special? It has a few key elements, and one of the key elements, it's really an academic and industrial mixture with a very intensive interplay, as you can see in the example of Justina, who spends most of the time at NANION, but still involved in the PhD program at Saarland University. Also, the consortium has a special employment policy that makes it a very open program. This is kind of endogenous to this EU-funded Marie Curie projects, because you are only allowed to employ PhD students that coming from a different country. So it avoids like this in-house PhD students that already did the bachelor and the master's and then continue as a PhD student. So this training program will really bring fresh blood into all the research groups. And the third point, and maybe the most important one, is there is a real exchange and a real collaboration. And one of the points that ensures this collaboration is the compulsory secondments, meaning each PhD student has to go to different labs for at least twice within the PhD period and learn new techniques or solve own questions with other techniques in different labs. And this results in a real exchange. And there's also a special relevance policy, what is unique to the relevance project. And this already started with the kickoff meeting, where we invited a lot of candidates, and it resulted in the fact that all the PhD students did know each other even before the project really started, and this already connected them. And I mean, with today's information exchange media, it's then easy to stay in contact, and there is really a lot of collaboration and exchange already within the PhD students. So this is these were a few introductory words about the Relevance project. Now I'm coming to the topical presentation, and well, I want to start very general, the concept of ion channel interplay, and I would like to start with some textbook knowledge, what is actually outside the red blood cell field. It's more cellular cardiology. And what we find in the cardiology is that we start with an electrical stimulation, well known as the ECG, and it is converted by ion channel activity, and in particular by calcium signaling, into the contraction, and this is what we know as the heartbeat. We can look at this on the cellular level. So you have two channels here displayed of isolated cardiac myocytes. These are cardiac myocytes from a rat, and you see whenever this red headlight electrical stimulation flashes, an electrical impulse or electrical pulse is given. And when the electrical pulse is given, you see in the transmission image that the cells contract. And in the right image, you see the same image in a false color version. And this false color version represents the calcium concentration in these cardiac myocytes. So whenever you see warm colors, you have a high calcium concentration. And if you see the cold colors like blue, the calcium concentration is low. And you see what is schematically shown above. You get an electrical stimulation, and the calcium signal is mediating this electrical stimulation into a mechanical contraction. And for the cell to be able to, well, facilitate this behavior, it indeed needs some ion channel interplay. And well, the very basic principle is that in the membrane of the cardiac myocytes, as visualized here in the scheme, you have voltage-activated calcium channels, and this calcium entry through these channels activates the next channel, the so-called ryanodine receptors, the calcium-activated calcium channels, and only the interplay of these two channels allows the cardiac myocytes to contract. And then to make the entire story complete, also the calcium needs to be extruded from the cells again. This is done by the sodium-calcium exchanger in the cell membrane and the Serpa pump in the endoplasmic reticulum. So what is very established and very normal in excitable cells, like in muscle cells or similar in neurons, the question is something like this, or do we have a similar situation also in red blood cells? And to give you a presentation about these interconnections, we like to start to introduce you to a few ion channels in red blood cells. The first one is the so-called Gardus channel. The second one is PSO1, and this part will be taken over by Justina. And then we have this CAF 2.1 channel. Well, I like to start with, well, just 3 main slides about the Gardus channel, and I'd like to start with like the historical order of this channel discovery. So well, initially it was not known that it was a channel. So Gardus in 1958 published a very small one-page paper indicating that with increasing calcium concentration, you get an increased potassium efflux in the red blood cells. And this was then called the Gardus effect. And well, I think it's quite funny. It was a one-page publication in BBA under the category of preliminary notes, but it was sufficient to name the channel after the author. And well, it took something like 23 years until the first really ion channel recordings of the channel that we now call the Gardus channel appeared. And these were published by Hamel as an abstract of the European Physiological Society meeting. And I think, to my best knowledge, these are the first traces of the Gardus channel published. Well, in the meantime, there appeared dozens of papers about single channel recording of Gardus channel, but it took another 21 years until the Gardus channel was molecular identified. And well, I still call the channel Gardus channel, but I think there are a number of different acronyms for the channel, well, in the literature. So SK4 is one of them, the gene name KCNN4 is another one, or if you stay in the ion channel nomenclature, the K calcium 3.1 channel. I mean, these are all the same channel, what I call the Gardus channel. So this paper by Hoffmann et al. Was published in PNAS in 2003, and another 12 years later, we had another milestone in the Gaudes channel story. So there was the first report published in Blood that where it was shown that a mutation in the Gaudes channel indeed resulted in a disease. So I think these are the key publications to sketch the career of the Gaudes channel, so to say. So what are the important properties of the channel for today's presentation? The one thing is there are plenty of different inhibitors for the channel. TRAM34 is one of the most specific ones. There are also Senica-POC, what went already into clinical studies to block the channels, but also the classical clotrimazole or caryptotoxin inhibit the Gaudes channel. So we have, in terms of channel inhibition, we have quite a variety of pharmacological tools. The activation of the Gaudes channel in the red blood cell induces a very unusual property in terms of other cell lines. This is related to the fact that the resting potential in red blood cells is not determined by the potassium, as in most cells, but it's determined by the chloride distribution. And this means if you induce potassium permeability by activation of the Gaudes channel, it has a tremendous effect on the membrane potential. This means if the Gaudes channel is activated, means the red blood cell that has a resting potential of about -10 millivolts immediately jumps to something around -70 millivolts. And we will come back, or I will come back to this property later in the presentation. And what is also important to know, what is the consequence of the activation of the Gaudes channel for the cell? So if the Gaudes channel is activated, obviously potassium can leave the cell. And the chloride permeability of the red blood cells is also very high. So because of electroneutrality, if potassium leaves the cell, also chloride leaves the cell. And well, then consequently, if KCl is leaving the cell, also you have a loss of water, and this means activation of the Gaudes channel results in a cell shrinkage, so the cell decreases the volume of the Gaudes channel is activated. So I think this is the background information about the Gaudes channel, and now I hand over to Justina to tell you a little bit about the PSO1.
Okay. So today's agenda will cover the following points. So I will talk about Nannion's contribution to relevance network. I will then point out the highlights of my talk. Then I will introduce you some information about PSO1 channels, tell you how we developed a method to record PSO1 channels in N2A cells, and then how we transferred the method to red blood cells. Finally, I will show you a novel PSO1 mutant in patient red blood cells, and I will conclude my talk with a little summary. Okay, as partner of the Relevance Network, Nannion Technologies have been involved in 2 work packages. The first one is diagnostic tools on red blood cells, with the aim of establishing new diagnostic means for rare anemias, using, of course, our automated patch clamp instruments. And red blood cells as treatment targets in pathophysiological conditions. And this is, for this purpose, we studied channel activities in patients, and we performed pharmacological screens on channelopathies. And to achieve these goals, we've used 2 patch clamp instruments in Nannion, which are PatchLiner, which is an automated patch clamp device capable of recording 8 cells at a time, and the SynchroPatch 384, which is capable to record 384 cells at a time. The highlights of my talk today will be the description of a patient with novel piezo-1 mutation and the patch clamp-based high throughput screening assay for piezo-1 activity. So here is a little introduction about piezo-1 channels. Piezo channels are members of a family of channels identified in 2010 as pore-forming subunits of mechanosensitive channels. Piezos are non-selective cation channels conserved among various species, and for example, the vertebrates have two distinct ion channels, which are piezo-1 and piezo-2. Today I will focus on piezo-1, which is widely expressed in the human body, especially in skin, flow-sensitive cells, endothelial cells, and most importantly for me, red blood cells. Piezo are large transmembrane proteins, and recently has been discovered the structure of the murine piezo-1 via cryo-electron microscopy, and the piezo-1 complex is now a trimmer shaped like a propeller with 3 blades organized around a central pore. So piezo-1 is believed to be expressed in the red blood cells because mutations of this gene are associated with hereditary xerocytosis, which is a rare autosomal dominant human condition characterized by stomatocytes and an impaired cation permeability. Stomatocytes are red blood cells with an oval or rectangular area here highlighted in the picture. And dehydrated red blood cells lack flexibility, and therefore they cannot squeeze through the small blood vessels in the circulation. Piezo-1 channels are supposed to sense shear stress and regulate cell volume in red blood cells. In fact, activating the piezo ion channel by mechanical stress or iodine administration promotes an influx of calcium ions, and this stimulates calcium-dependent signaling pathways in the cells. For example, the opening of Gardus channel. The resulting efflux of potassium channels, ions, through the Gardus channel leads to a loss of chloride ions and water, as Lars previously explained as well. Now I will talk about our data. So a patch clamp assay to measure piezo-1 currents was first established on the patch liner in Neuro2A cells. Neuro2A cells are mouse neuroblastoma cell line endogenously expressing piezo-1 channel, and these were kindly provided by Gary Levin from Berlin. Cells were recorded at holding potential of minus 60 millivolt in physiological solutions, and piezo currents were stimulated by administration of iodine at a final concentration of 20 micromolar. The graph shows 2 example sweeps or current traces, each of 500 milliseconds, and the dark blue trace represents here the baseline current and before iodine addition, and the light blue trace is recorded after iodine addition and shows an increase of 20 picoamperes in the current compared to the baseline. The second graph here displays the minimum value of the current for each sweep plotted over time during the experiment. This is what we call IT plot, and clearly shows an activation of the current upon iodine administration. The same result can be appreciated also in the scatter plot here for 12 cells, and you can also see how heterogeneous is the response of these cells to iodine 1. It is because of this heterogeneity in the Yoda response that we transferred the method to the Nanion high-throughput patch clamp instrument, the SynchroPatch. The recording conditions were kept the same, except for the voltage protocol here, that from now on will be a ramp ranging from -100 to +80 millivolt, and at an holding potential of -60 millivolt. And the Yoda concentration was reduced to 10 micromolar because we saw that it was already enough to activate the current. Here is a screenshot of the data acquisition and analysis software of the SynchroPatch 384PE. So the upper left panel displays the 384 wells sorted by color, and this is based on the seal resistance values. All the wells with the seal resistance above 500 mega ohms are here highlighted in green. Then the bottom center highlights the raw traces of a selected cells, and you can appreciate in black the trace before Yoda addition, so the baseline current, and in red the trace which is recorded after Yoda administration. The red and green vertical lines here are what we call the cursors, and they are useful, so they are our region of interest for the analysis, respectively at -100 and +80 millivolts. While on the right side here, raw data traces of 16 selected wells are displayed. So when moving to the online analysis view, we can see the time course of the experiment, and the compound addition is here highlighted with different shades of blue. We have the Yoda addition, which is the piezo-1 agonist, and then gadolinium chloride, which is a non-selective blocker of piezo currents. Okay, Yoda-1 responder cells were selected according to strict quality control filters, such as stable resistance values over time, and a block by gadolinium chloride more than 20%, and a Yoda response higher than 20% as well. And in one example chip, 37% of the cells were considered as valid cells after our quality control filters, and of these valid cells, 60% of the cells were considered as Yoda responders. And you can see here what we used as Yoda responders, an example of Yoda responder and an example of Yoda non-responder. Okay, when moving to statistics, our approach here consisted of evaluating two groups of cells independently. So Yoda responders on one side and all cells, so responders and non-responder cells on the other side. And statistical analysis showed a highly significant increase of the current whenever 10 micromolar Yoda-1 was applied. Okay, in the next experiment, the physiological external solution was exchanged to an NMDG-based solution in order to further characterize the nature of Yoda-1 induced currents. Taking advantage of this high throughput system, so the SynchroPatch, we could compare the experiments with the two external solutions in the same chip. Now the graph on the right reveals that the inward current elicited in NMDG-based solution is reduced, and this is demonstrative that our channel, so what we are studying as piezo channel, does not conduct NMDG ions, and this is consistent with the literature. Okay, an optimized high throughput method was then applied to red blood cells. Blood samples were flashly obtained from a healthy donor and suspended in calcium-free external solution. The voltage protocol, solutions, and compound were kept the same, except for holding potential that for red blood cells is set to -30, and a special chip resistance, which has to be adapted to the tiny size of the cells. Again, the trace view The trace view of the acquisition software highlights a great success rate, with more than 17% of the cells showing a SIL resistance higher than 1 giga, as you can see here in green. The activation of the current by IODA-1 and subsequent block by gadolinium chloride are better appreciated in the online analysis view here. So as for N2A cells, only the red blood cells that passed the quality control filters were considered for the analysis. In one chip, 58% of the cells were considered as valid cells, and we had to consider strict values for the SIL resistance, stricter than the red blood than the N2A cells. And of these valid cells, only 33% were considered as IODA responders. And here is an example of IODA responder and non-responder. We investigated as well a putative Gardus channel contribution to the IODA-1 activated currents in red blood cells. Basically, we performed the same assay in two different experiments. The first experiment was done with in the presence of TRAM34, a specific Gardus channel blocker, and the second one was done in the absence of TRAM. So we can see from the graphs here that at positive potential we'd have there was no significant change induced by TRAM in the currents, while at negative membrane potential there was a slight significant increase in the current amplitudes. Since this change is opposite to putative contribution of Gardus, we can say that in our conditions a participation of the Gardus channel is excluded. Okay, now the funny part, the patient. We used the assay developed on the red blood cells and the N2A cells to measure patient current in red blood cells. So the patient is an Italian man with no evidence of jaundice or anemia or splenomegaly, and was studied at the age of 43 years for compensated hemolysis and iron overload. So the presence of stomatocytes in this peripheral blood smear and the observation of a different red blood cell deformability, which is displayed with a dotted line compared to the control, suggested that there's a mild form of hereditary xerocytosis. Here is a schematic representation of PSO1 gene and its protein. Highlighted in pink, here are the exons in which mutation has been already described, and they correspond to the orange areas here in the protein representation below. The novel mutation sits in this region here, and is highlighted also in yellow in this structure on the side. This, the region in which the mutation sits is called anchor region, and this is supposed to affect the gating of the ion channel. Okay, now we have some data. So here are examples of shipped control and patient red blood cells, respectively. And we can see that already at the baseline conditions, the values of the current are indicating a highly significant difference between shipped control and patient, both at positive and negative potentials. This difference persisted after application of IODA-1, And as well, we consider the YODA responders and all cells, so responders and non-responders. So in line with previous reports of PSO1 mutations in red blood cells, and considering also our data, so both baseline currents and YODA-induced currents, we suggest that the mutation that we are investigating, the novel mutation, is a gain-of-function mutation. And the structural localization is also supporting this conclusion. So now I get to the conclusion. I've shown that Nanions patch clamp instruments are valid tools to study PSO1 channels via chemical stimulation in cell lines as well as primary cells. I've presented the first electrophysiologic screening ever performed on red blood cells, revealing a functional difference in patient currents and also when compared to healthy control. And I've provided a putative approach for detecting functional channel mutation, not only in red blood cells, but this is also applicable for other primary cells like myocytes, neurons, or epithelial cells. So now I will hand over again to Lars, and thank you for your attention.
So now we have heard about the Goddess channel and about PSO1, and now I'm continue with CAF 2.1, and this is one of the channels that is really hard to grasp. So what is the indication? There was a paper published in 2002 in Blood, where there is really molecular biology indication for the channel. So this particular band, so in the B version of each sample, there is a peptide that specifically binds to this channel, and then the band in the Western disappear. And there's also lots of pharmacological indications with a toxin that's called omega-acatoxin TK, what is supposed to be specific for the CAF 2.1. And here we have just what we see here is the fluorescence intensity corresponding to the intracellular calcium, and the blue bar gives the control conditions, and LPA, lysophosphatidic acid, stimulates the red blood cells, what is believed to activate this CAF 2.1 channel. So you get a measurable increase. So we can skip the gray bar. It just shows that there is, well, in this particular channel activation, there is no PKC alpha involved. But if we add this agatoxin while stimulating this LPA, you really get the blockage of the calcium. And therefore, we believe this channel is really there in red blood cells. Although nobody managed to measure the channel by patch clamp so far. And well, also Polina Petkova-Kirova, who is one of the attendees, tried very hard to show the activity of these channels in red blood cells, but did not succeed. So well, what I like to come now to is the interaction of all these channels. And the motivation to go into this is a disease, what is called Goddess channelopathy. And it's based on a mutation of the Goddess channel. And well, we also characterized the channel by the patch clamp. So this was where our patch liner came into operation, and it was again operated by Polina. And there is a point I like to make, because if you look into the literature, most references in terms of Goddess channel mutations points to hereditary xerocytosis. And as we have seen in Justina's presentation, if the channel has xerocytosis, this osmoscan, the curve that is plotted here, is shifted to the left. But what we see in the patient of this particular mutation printed here, R352H, there's no left shift of this osmoscan. And therefore, we believe that the Gardus channelopathy should be regarded as a separate disease. So what was now very interesting when we investigated the cells of this patient was that the cells, the red blood cells of this patient showed increased intracellular calcium concentration. So here you see example images, and here below you see the statistical analysis. So this is a particular patient who has a de novo mutation of the Gardus channel, and this is his daughter carrying the same mutation. And both patients have this increase in calcium, and this is something we couldn't explain ourselves because we got a gain of function mutation in the Gardus channel. What is activated by calcium but not permeating calcium? It's a potassium channel. So we wanted to study this phenomenon, and to study the phenomenon, we used the pharmacological tool, what is called NS309. And we use this tool because, well, the Gardus channel is calcium activated, so we would need to increase the calcium to activate the channel, but at the same time, we wanted to use the calcium as the readout to see how the Gardus channel interacts with the calcium. So with the NS309, we have a tool that we can increase Gardus channel activity without increasing the calcium. And we did some experiments by flow cytometric analysis. And if we stimulated the cells with NS309, we got a population of high calcium cells. This is what we call the responding cells, in contrast to the non-responding cells. And here in the diagram in the left, we plot the number of these high calcium responding cells in dependence of the NS309 concentration. And we see at concentration of 100 micromolar, we get a really decent response of the red blood cells, so a number of something like 25% of high calcium red blood cells. Now we ask ourselves whether this is really caused by the Gardus channel, and we use two different Gardus channel blockers, caryopytoxin and this STRUM34. The blocker alone doesn't do anything, but if we simultaneously stimulate the cells with NS309 and at the Gardus channel, we got for both blockers exactly the same decrease in the calcium, high calcium cells as you compare with the high concentration. So we really believe that to a big part, the high calcium cells are really caused by the activity of the Gardus channel. But well, so far we don't understand why. So we did the next round of experiments. So what you see here is again what we've seen in the previous slide, just a different measurement. So roughly 30% of cells respond to NS309. And then we used again this omega-agathoxine TK, what is supposed to be specific for CAF 2.1. Well, the drug alone doesn't do anything, and well, if we simultaneously or if we pre-incubate with the inhibitor and then stimulate the cells, we got a decrease in the high calcium population. So we got the idea, okay, this CAF 2.1 may make a contribution to this high calcium cell, but it's not the sole source of the calcium. So then the obvious hypothesis was, okay, maybe the other contribution comes from the piezo-1. So we did the same experiment, but not inhibiting the omega-agathoxine TK, but we inhibited the piezo-1 channel. And then the big surprise was we could not reduce the amount of high calcium cells, but we got an increase in the high calcium cells. And what was very surprising on the first view is not so surprising if you have a closer look. And I try to explain you what we think is happening. And to do so, we just look at the channel, or the CAF 2.1 channel activation in general, independent of the red blood cells. So if you look at these voltage activated channels, you need to consider three different states of the channel. So you have the closed channel, and the closed channel needs a depolarization to be opened, but then it's not directly converted into the closed state. It's first inactivated, and then needs a hyperpolarization to be in the closed state again. So this is a general property or a general behavior of this CAF channel. So how is the situation in the red blood cells? So we believe this hyperpolarization could easily be done by the activation of the Goddess channel. Because if the Goddess channel is activated, we have to jump the membrane potential from -10 to -70. So it's exactly a hyperpolarization. But how to realize the depolarization? So the idea is the depolarization we see if the Goddess channel closes again. And this can, for instance, be stochastic closures. And if you consider, if you consider that the number of Goddess channels in the red blood cell is very, very low, so possibly a single-digit number, you can imagine that if you consider the stochastic behavior of the ion channels, you come in the situation that at a certain time point, all Goddess channels are closed, and then you get the depolarization. So this is at least our current and published hypothesis. So how this now interplays with the Goddess channel activity? So if the Goddess channel is activated, for instance, by the NS309, we get this membrane potential change, and this membrane potential change, as I just explained, may activate this CAF 2.1 channel. At the same time, you get a water loss due to the activation of the Goddess channel, and this may act as a mechanical stimulation of the cell, and this would activate the piezo channel. But once the piezo channel is activated, and as you have seen from Justina's talk, piezo is a non-selective cation channel. So if the piezo channel is activated, means the membrane potential, because it's non-selective, the membrane potential goes close to zero. And therefore, this membrane potential change here is kind of shortcut. So functional, the activation of the piezo, functional, the activation of the piezo blocks the CAF 2.1. I mean, it's not a direct blockage, but by clamping the membrane potential. And this, and then if we block the piezo, this counterproductive activity of the piezo can't act on the CAF 2.1, and we get a higher number of high calcium cells. So this is at least our current hypothesis based on this NS309 experiment. Now we like to go back to these patients with the Goddess channel mutations. These patients, or this mutation, is a gain of function mutation, and therefore we would expect if the Goddess channel is more active in these cells, we would see something similar also in these patients. The problem is that the higher activity of the Goddess channel is not as pronounced as in our NS309 experiments. So therefore, we did experiments over a time course of 24 hours. So we loaded cells with the calcium dye and incubated them with the different inhibitors. So without incubation, so these are control conditions, we get a certain number of high calcium cells. And again, we used two different inhibitors of the Gardus channel. We got a reduced number of high calcium cells. We also get a reduced number of high calcium cells if we inhibit the CAP2.1. But if we inhibit the PSO1, we get an increased number of high calcium cells. So the patient mutation reflects exactly the pattern of our NS309 experiments. Well, unfortunately, this is just one measurement, so we don't have error bars, and we can't test for statistical significance. But I think the pattern is in total agreement with our NS309 experiments. So this is already the end of my explanations, and I've just a few take-home messages left. And there are indeed busy channels in the red blood cells. The high throughput patch clamp has a diagnostic potential in hematology. The Gardus channel is not, as believed for a long time, a relic, but it's a vital channel. And red blood cell channelopathies turn out to be quite complex, or to have a quite complex mechanistical interplay with different ion channels contributing, although only one channel has a mutation in the first place. So I'd like to thank you for attending this webinar, and I think I'll pass over to Gesa again.